Okay, this is year 12 biology, and we're learning 4.5, the cell cycle. Well, we usually represent the cell cycle just by drawing a circle. So you start with, let's choose black. You start with a circle. And what you want to do is kind of draw a Mercedes-Benz logo in the middle, right? So you have a circle and then you have three lines in the middle. Mercedes, Mercedes-Benz. And what you want to do is in the segment, in this segment here, you want to draw a line like this that divides the area like this. And we start with G1. So that's our first phase in the cell cycle. After G1 follows S phase, that stands for synthesis. And after the S phase, we have G2. Okay. Now, this phase is what we call mitosis. So this has PMAT, PMAT, prophase, metaphase, anaphase, telophase. You know what they are. After learning this basic cell cycle, we need to know a few checkpoints. So in another color, we use green here. We're marking out the checkpoints in the cell cycle. There are 3 points in a cell cycle. The first one is in G1, and the second G2, and the last one is in the M phase, mitosis. I think you only need to know where they are, but not as much what they do. But I'm gonna include that for the purpose of this video. Well, G1 checks for DNA damage, nutrient levels, growth factors, cell size. G2 checks for the amount of DNA, whether the replication went well, and if there is any mutation in the DNA. And the reason for doing that is following directly after the G2 phase is mitosis, and mitosis is where we need replicated gene already prepared and ready to go. So you need to make sure that the replicated genes don't contain any mutation. And the cell cycle last checks for mutation and stuff in the mitosis phase. So that's the cell cycle. Now, related to this topic, we need to know a few factors that also regulate cell division. There are two types. There's internal and external. So internal has three and external has four. The first internal factor is the size of the cell. Is it too big? Is it too small? Right? If it's too small, it can't replicate. If it's big enough, it can replicate, it should replicate. So that's what cell size means. And the remaining two are kind of similar. This is the second one is regulatory genes, and the third is regulatory proteins. Now, they're similar in that the regulatory genes code for the regulatory proteins. So with either of these gone, the regulation won't occur because the gene codes for protein, and if the protein isn't synthesized, the regulation won't happen. So this is pretty clear. Now external has four. The first one is nutrient, pretty easy, nutrient. Now the second and third, they're kind of related, anchorage and the density. Now, the full name is anchorage dependency. This is when a cell only divides if it's anchored onto something. So if it's just floating around, like a fish, like jellyfish in the ocean, just floating around without an anchor, it won't divide. To be able to divide, it needs to find an anchor, like attached to something, to be able to divide. And that's one of the properties of cells. And the third one is density dependency. This is when a cell knows what's around it, right? It knows the environment around it, whether there is too much density or too little density. An example of this is our skin cell. Now, our skin cell could just divide and divide and divide, and it will form this huge lump, this saggy skin, saggy sack of skin if they divide too much. But in real life, they don't. They're perfectly fine, like perfectly tight. And the reason our skin does that is because it's thanks to the density dependency of this property. This is when cells can detect if there is too much stuff or too little stuff around it. If there's too much, say uh, too much stuff, it will stop divide. If there's enough space, it will just keep growing and divide. And last one is growth factors. Now this one is kind of interesting because it's growth factors and in the bracket hormones. So growth hormones. Anyways, these are the two types of factors that regulate cell division. And speaking of growth factors and growth hormones, we need to discuss that in the right now. Right now. So the growth hormone is released by the pituitary gland. If you study year 12 biology, you will know already that pituitary gland controls pretty much everything. It secretes a lot of the stuff that our body depends vitally on. And in this case, it releases the growth hormone. So let's just say the pituitary right here. Ugh, that's disgusting. I just use the letter to represent P, pituitary. And it releases GH. This stands for growth hormone. And this goes to the liver. So you have your juicy chunk of liver right here. Try my best. Liver. So the pituitary starts with pituitary, releases growth hormone, which goes to the liver. The liver processes this message because it's a it's a chemical message that tells the liver to produce growth factors, GF, not for girlfriend, growth factors. And this could go all places. Uh, if you are in puberty, you could go to your bones and tell them to grow and divide. That's why you grow taller when you're a teenager, right? This could go to your bone cells, bone tissues, and this could also go to your muscle cells, right? You also gain muscles when you're a teenager. So, overall summary, last time summary, growth hormone goes to the liver. The liver secretes growth factors, which goes to the body tissues and tell them to divide and divide and divide. Next, we're going to learn an example of regulatory genes and regulatory protein. Uh, so this is an interesting relationship. We need to learn about one. First one is CDK. CDK is a hormone. And the second, we have cyclin. Cyclin is a protein. Actually, mistake. CDK is an enzyme, and cyclin is a protein. So what you know about CDK is it's an enzyme that's always there. Its level, its concentration in our body stays constant, no matter what the no matter what phase the cell is in, whether it's in G1, S, G2, M, doesn't matter. CDK level stays constant. Now, cycling level, on the other hand, it could rise and drop, and it depends on the phase that the cell is currently going through. So, what do these two and what's their relationship? Well, cyclin binds to the CDK, binds to this enzyme, and together they form a complex that tells a cell to pass a checkpoint, right? So this could, just for this example, this particular CDK and this particular cyclin together form a complex that tells the cell to, get through the G1 checkpoint. So when the cell approaches G1, the G1 phase, well, the end of the G1 phase, you see a rise in the concentration of cyclin at that time. But the concentration of CDK will still stay constant. But since the cyclin level is rising, right, uh, a great number of cyclin is binding to the CDK, forming that complex, which instructs the cell to get through that checkpoint. So, with enough accumulation of the cyclin and the constant level of CDK, the cell gets through that checkpoint. And what happens then? Well, you see the cyclin level goes down. So, this could be the start of the S phase, and this could be the one end of G1. When it gets through that checkpoint, it drops because it's not needed anymore. The last dot point, we need to learn the two types of, actually, no. Actually, yes. Two types of cancer. Um, Two types of cancer, A and B. A stands, not A, no, doesn't stand for anything. A is just benign cancer. Benign means it's harmless, right? Benign. It's harmless. And the second, we have malignant. The major difference between these two is benign doesn't spread. If you have a benign cancer in your thigh, hopefully you don't. If you have benign cancer in your thigh, the cancer will just stay there, right? It doesn't spread. It doesn't break off your thigh muscle and go around the body and start a new colony in your brain. A benign cancer is, well, supposedly easier to treat than a malignant cancer. Malignant cancer. Now, this is the tough stuff. All right, this is the annoying cancer because if you get this in your thigh muscle, and if it's a malignant cancer, it could break off. Right, it could break off and travel in your bloodstream with your blood and lodge somewhere else in your body. That's why cancer spreads, and that's how cancer spreads. It breaks off and travels with the bloodstream and sits somewhere else. It could be in your lung or in your brain, and it's real annoying. But anyway, benign is good. I mean, it's not as harmful, but it can be harmful. And malignant is definitely really, really bad. So that's all. That's unit 2.5, the cell cycle. Thank you.